Is the nervous system pretty important? Do you know one way you can tell your nervous system is important? It controls your body. It controls every cell in your body. And a conservative estimate that I found from the Smithsonian Institution says the human adult has 37 trillion, with a T, cells. Some estimates go up to 100 trillion, some are as low as maybe 15 trillion. Basically, when you see all those zeros, that's a lot. And if your nervous system is able to do that, that's pretty impressive. Because those cells have to be in the right place at the right time, knowing when and when not to divide. Some cells have to know when to die to get out of the way. This impresses me because I have 2 granddaughters that I can't control when I babysit. 37 trillion. But even though I can't control my granddaughters, our nervous system can accomplish some really important tasks. Not the least of which, your nervous system collects information. Your nervous system is like a detective, bringing clues in to try to figure out what's going on. So receptors giving you information about your external environment bring information to your brain. Receptors about what's going on inside your body brings information to your brain. You have sensors that tell your brain about the contraction state of your muscle. You have receptors from your inner ear that tells your brain about your position in space. You have receptors in your inner ear that tells your brain if you're moving, if you're accelerating, or if you stopped. That's pretty impressive. But all that information comes into your brain where it's processed. It's integrated. Your brain says, what does all this mean? What does it mean relative to the homeostatic condition of our body? Our temperature is too high. Our skin temperature, we're hot, we're sweating. We're doing that to lower our temperature, but in the meantime, we're also losing too much water, so we're dehydrating. We need to make the idiot thirsty so he'll drink. All this information is being processed so that you can have a response to try to integrate all of these 30 trillion cells, all of these physiological parameters to maintain everything in balance, to maintain homeostasis. And you want to maintain homeostasis and protect yourself even when you're stupid. And we're going to talk about how your body can do this without having any conscious input. That's going to be in our last chapter on the autonomic nervous system. So that's a lot. That's a lot of activities that go on. And when you look at the nervous system, it is a complex system. And so, like all good scientists, I have to categorize, organize, find pigeonholes where I can make sense of all this stuff. So the first thing that we're going to do is we're going to break down our nervous system into 2 major categories. The processing centers and the transmission centers. So this is the CNS and the PNS. NS is nervous system. What does the C stand for? Central nervous system, your brain and spinal cord. This is the information integrating part. PNS, what's the P for? Peripheral nervous system. You're going to have these cables of nerve cells that are called nerves that are coursing from the spinal cord, from your brain out to the body. And outside the central nervous system where you have nerve cell bodies, those are going to be called ganglia. There's one you probably already know near the spinal cord. It's called the dorsal root ganglia. If you've heard of one, you've heard of that one. We're going to talk more about those as we talk about our nervous system cells. But when we look into PNS, we can break the PNS down into the transmissive lines, whether information is coming to the brain or information is going away from the brain. If it's coming to the brain, it's sensory. How does that feel? What did we touch? Is it hot? Is it cold? Am I thirsty? In our sensory division, we can break down into somatic and autonomic. Somatic is something that you're consciously aware of. That's heavy, that's light, that's hot, that's cold, that's blue, that's red, that smells nice, that stinks, that's too loud, not loud enough. You're squeezing too tight. Those are all senses a part of the somatic sensory system. Versus information we're getting from our internal organ. Blood pressure's too high. Heart's beating too fast. We're dehydrated. Sodium concentration is too low. pH of the blood is off. All of that is subconscious information. That's part of our autonomic system. And if you learn somatic and autonomic, guess what? On the motor side, we have the same thing. In our motor division, the somatic, soma meaning body, the somatomotor system controls skeletal muscles. Whereas our autonomic, which to me sounds a lot like automatic, involuntary, that's going to control cardiac, smooth muscle, and your glands. You're not going to be aware of those things happening, but it's so critical to control those as well. That's a big system. There's a lot going on. And there are a lot of cells involved in making all of this happen, to bring the information in, to integrate the information, to support the cells that transmit the information. But then all of the ways in which this information is utilized through the motor division, that's going to be important too. We've got those reflexes from the somatic division when you touch the hot stove. But then we have these other reflexes that happen in our autonomic system, one of which we call the fight or flight reflex. And we're going to cover that stuff at our last chapter. I believe that's chapter 14. And that'll be on our final exam. So as you look at all of these divisions and all of this breakdown, you know I like pictures. And so here's a picture that does the same thing. Central nervous system in the middle, peripheral nervous system around it here in this block. Our sensory input, external environment, that's our somatic. Then our autonomic coming from our internal organs. Here's our somatic motor division, skeletal muscle, autonomic division, smooth muscle, cardiac, and glands. Now this subdivision of autonomic into sympathetic and parasympathetic, that's chapter 14. We're not going to cover that certainly in this first part, this chapter 12 part, but we'll get that in the nervous system as a whole in chapters 13 and 14 for the final. All right. Any questions about these big divisions? We're going to start off looking at the cells of our nervous system and what these cells do, how we can categorize them. We're going to look at their structure. We're going to look at their function. And of the two major categories of cells in the nervous system, we're going to start with the stars of the show. When you think of a movie and the actors and actresses that you see on the screen, these are the neurons. They transmit the signals. They signal each other, they signal muscles, they signal glands. So they're the excitable ones. But the one thing that neurons can't do that most cells are capable of doing is neurons cannot divide. They're what's called amitotic. Anytime you put a or an in front of a word, what does that mean? Not or without. So these cells are without mitosis. They can't divide. Once you lose a neuron, you don't replace it. So when you watch a movie and you look at the credits at the end, do you just see the names of the actors and actresses? Or do you see all the names of the people behind the scenes that made it happen? Well, the behind the scenes players are the glial cells, or you can refer to them as neuroglia. This is the supporting staff, the protecting group. And just like when you look at the credits, the actors and actresses make up a minority of what you see in the credits. Neurons in the nervous system, what percent of your nervous system would you think neurons are? I mean, these are the ones that send the signals. Without your neurons, you wouldn't be able to function. What percent of your nervous system would you think accounts for neurons? 10%? 12, 40, 30? What do you think? In fact, it's 20%. 1 out of 4 of your cells are neurons. The others are the supporting cells. And again, that's much like you see in the movies. Now, to say that neurons are more important, I mean, they're clearly important because we all have all these cells supporting them, but without these glial cells, your neurons wouldn't be able to do their job. We have some pathogenesis that basically attests to that fact of how important the glial cells for nervous system function. So, let's look at the neurons first. And let's look at the parts of our neuron, the cellular part. Because each of these parts is going to function differently. And they're going to function differently because of their structure and because of the proteins are present in their plasma membranes. So here we have an illustration of 2 typical neurons, our most common types of neurons in our body. We can see the nucleus is associated with the cell body. And then you have a lot of these processes that come away from the cell body. Collectively, these processes are called neurites. But some of the neurites are numerous and short, and one neurite's going to be really a lot thicker and longer. Well, our short processes that are off of our cell body, these are called dendrites. And these dendrites are responsible for bringing signals, stimuli from other neurons, bringing them to the cell body. Now, an analogy I like to use with neurons is your own nervous system. We have sensory nerves bringing information to the brain. The brain integrates that signal and then sends information out through your motor neurons. So in this sense, our dendrites are like the sensory nerves bringing electrical information to the cell body. Now, what's in the cell body, the thing I want you to remember, the function of the cell body, is that it integrates the signal. Highlight that word, integrate. Because when we're talking about signals, we're talking about changing in membrane potential. And we're going to change the membrane potential all along all of these dendrites, and we're even going to have synapses on the cell body itself. So we're going to have a lot of changes. We're going to have a lot of ions moving around. And all of those ions changing places are basically going to flow into the cell body, flow around in the cell body. And the cell body is basically going to add them all together, mix them all together. And whatever that change of membrane potential adds up to be at the very beginning of our axon, this long process that's sending information away. Whatever that membrane potential is at that point, that determines if that neuron's going to send a signal or if it's not going to send a signal. So ions change place all around the dendrites in the cell body. All of that added up at the beginning of the axon that's called the axon hillock. That's going to be the trigger to whether we send an action potential down the axon or not. Because dendrites bring information to the cell body, but the axon sends an action potential away from the cell body. Now, once that action potential possibly starts, it's going to go all the way down the axon until we get to the axon terminals, which we know are our synaptic knobs. We have a synapse. We already talked about all that with muscle. We're not going to spend a lot of time on the end part in the synapse. We're going to spend a lot more time on everything else, especially this first part. But before we leave, I just want to point out this term nerve. You'll see this term a lot. A nerve is a collection of axons. May have some dendrites mixed in some of these, but principally a collection of axons, bundled all together in the body. And when we wrap them up with connective tissue and bundle them together, you can visibly see these in a lab, and you call that a nerve. So here's an example of one of our nerves. This is a peripheral nerve. Here's our spinal cord. And in our peripheral nerve, we'll have some sensory fibers. We'll have some motor fibers, not that you can distinguish them in just a examination. But you can see we've wrapped a lot of connective tissue around all of our axons. And we have 3 different levels of wrapping. We can wrap individual axons with connective tissue. And that particular wrapping we call the endo, which means inside. Neurium is just referring to the nerve. So the connective tissue that wraps each and every axon is called the endoneurium. But we don't stop there. Much like we see in muscle, we take these axons with their connective tissue and we wrap them into these bundles called fascicles. And we wrap these fascicles with connective tissue, which we refer to as the perineurium. Did you get this when we did muscle, in the lab with muscle? And it's the same thing in nerve. Y'all just starting nervous system in lab? So we've kind of caught up. Good. Now, the third level of connective tissue wrapping is you take all of the fascicles, wrap them together with connective tissue, and that's called the epineurium. The epineurium is the outer part of the nerve. So again, just a little bit of anatomy as we're building our picture of what the nervous system is going to look like and how all of these cells function together in our nervous system to control our body and help us maintain that H word that I've said a number of times, homeostasis. So we have our 3 basic parts of any neuron. The dendrites, information in, cell body, integrating the signals, axon, sending information away. But the number and the position of these processes can help us classify our neurons based on their processes and how they look. And so here you see below pictures of 3 basic types of neurons and the names of these. So the most common type of neuron is what you see in the illustration on the left. Multipolar neurons have a number of processes coming off the cell body, but most of them are going to be short dendrites with one long axon leading away. That's the most common form of our neuron that we have in the body. But we also have other looks, not the least of which is one here in the middle. This one in the middle is named not because it can't make up its mind, because it has 2 main processes extending away, and so that's the bipolar neuron. That's pretty straightforward, the reason you got that one. But then our third type, just based on its cytology, is the fact that we have this one large cell body and there appears to be one process extending away from it. Now, your book likes to use this word unipolar. I prefer to stick pseudo in front of it because you've got information coming in and out and there's really 2 highways there. So what does the word pseudo mean? Fake, false. So it looks like one, but it's really 2. And so this is our pseudo unipolar. So this is how we look at the processes, the neurites, and we classify the cells based on how they look and the number of processes leaving the cell body. Now this is also going to relate to different function, and we're going to get to that in the next couple of slides. So the bipolar, this one, has 1 dendrite and 1 axon. In this illustration, we don't have an arrow to let us know which direction the information is moving. We would need that information before we would know which is which. Any other questions? So we categorize them based on the number of processes. Now we can categorize them based on their function. So we've already talked about sensory neurons, bringing information to the central nervous system. And so here's that word I sort of introduced last week, afferent, A-F-F, A meaning arriving. So sensory neurons bringing that information to the central nervous system. Now again, these neurons are typically going to be that unipolar, pseudo-unipolar type. So most of that spinal nerve is going to be the dendrite bringing information to that cell body. And the cell body of that sensory neuron lives in a ganglia, which is a collection of cell bodies in the peripheral nervous system. And that ganglia is going to be at the dorsal part of your spinal cord. And so guess what we call that ganglia for the cell bodies of our sensory neurons? The dorsal root ganglia. And you'll see that one in lab if you haven't already. Now, our motor neurons, these are going to be our multipolar. This is one of the reasons why they're one of the most common types and forms. Our motor neurons are efferent, E for exit. They're sending information away from the central nervous system going out to the effectors, including muscles and glands. The two things that actually get the work done in the body. Now, these are going to start in the ventral root, the ventral horn of the spinal cord, exit the ventral root and get collected into our peripheral nerves. Now, in some instances, we integrate the signals. We can actually diversify where the information goes with neurons that are called interneurons. And interneurons simply connect other neurons together. And one of the places we see this the most are in our spinal cord with our reflexes. You have a sensory neuron come in from your fingertips. When you touch that hot stove, the information comes in very quickly. But rather than send information directly to your brain, you have an interneuron that connects from that sensory nerve to the respective motor nerve so that when you touch that hot stove, you move your hand away rapidly. And it's that interconnected nature that allows us to have things happen very, very quickly. So here in our illustration, we have a sensory nerve bringing information in. There's the cell body in our dorsal root ganglia. You can also see it illustrated a little bit more histologically down in this lower picture. But the information comes into our central nervous system. There's an interneuron that's transmitting the signal from the sensory to the motor cell body. There you see the dendrites. And now the axon of that motor neuron, the multipolar neuron, exits the ventral root and it goes out to the effector muscle. That would kind of be an example of a reflex. But do you see how the three neurons are working together? Sensory, inter, and motor. So we've described them based on how they look, their processes. We've described them based on function. Now let's look at the supporting cast. The neuroglia, 80% of what we have in our central nervous system. There's 80% because of how much they have to do. And there's a lot that these neuroglial cells do, in addition to protecting, supporting, nourishing, and in some instances guiding neurons when they repair, which principally is only going to happen in the peripheral nervous system, but it does happen. And so we're going to take our neuroglial cells and we're going to talk about them. There are basically 4 in the central nervous system and 2 in the peripheral nervous system that we're going to spend time talking about and getting to know. So, I love pictures. You know I love pictures. And so in this particular illustration, which includes 4 of the main neuroglial cells in the central nervous system, we're first going to start with the little green Martian looking thing. And that green cell that's illustrated is an astrocyte. I guess I kind of like it because it's cool that they named a cell after the World Series champions, right? It's pretty cool. No, they had the name for a long time because astrocytes have a number of processes. So an early histologist said, wow, this looks like a star. Astro meaning star. And so all of these processes from the astrocyte connect to the cell bodies of our neurons, but they also connect and cover capillaries. These are referred to as perivascular feet. And so our astrocytes are essentially going to form what you probably have heard of as the blood-brain barrier. Have you heard of that before? The blood-brain barrier is a protective barrier against infectious agents. And so the astrocytes are actually going to screen any material coming from the capillary, oxygen, carbon dioxide, glucose. It's first going to pass into the astrocyte, and if the astrocyte deems it's good, it's going to pass it on to the neuron. Likewise, waste products are going to be transferred from the neuron through the astrocyte back into the bloodstream. That's a pretty good deal, right? Infectious agents not going to get past the astrocyte. But it creates a problem also, doesn't it? If someone has a central nervous system infection, especially a bacterial meningitis, how are you going to get antibiotics to the brain? You're not going to get them past the astrocytes. So while it's a great protective measure, it also presents a medical problem in treating people that have central nervous system diseases, because you can't get medications past the astrocytes. So the astrocytes, blood-brain barrier, protective. They nourish, they support the neurons. So the next cell I want to look at are the microglia, these little blue ones. The microglia because they're the smallest of our glial cells. But they're not least important because the microglia are also protecting your neurons, because microglia are the macrophages of the central nervous system. And what do macrophages do you remember? They're the vacuum cleaners. They go around and clean up debris. They get rid of pathogens. They phagocytose them, gobble them up, eat them, and destroy them. So the microglia protect the central nervous system. Wow, so far astrocytes nourish and protect, the microglia eat bad stuff and protect. Are you getting the message of what these things do? Now the third type you probably have never heard of before. These are called the ependymal cells, and you can see them here in pink at the lower right. Ependymal cells line cavities in your central nervous system, and these cavities are called ventricles. You probably only have ever heard of ventricles relative to the heart. But ventricles are these open spaces in your central nervous system that are filled with a fluid. This fluid is called cerebrospinal fluid. You ever heard of someone have a spinal tap? They're taking some of the cerebrospinal fluid out, looking for infectious agents. But this fluid flows through these cavities of the central nervous system and actually flows around the brain and around the spinal cord. So this fluid almost acts like a moat around a castle. So any infectious agent that's trying to get into the brain directly through the membranes or the meninges, it's going to hit this flowing river of cerebrospinal fluid and be washed out before it can get to the actual nervous system tissue. But the other thing with this fluid, since it's in this confined space around the brain and spinal cord, it acts like a hydraulic shock absorber. And so the fluid keeps your brain, most of the time, from slamming against your skull. Unless you've got a football helmet on and you get jacked up, right? So once again, we've got fluid that's protecting the central nervous system. Well, the fourth type of neuroglial cell that again is here in light blue with all of these processes is a cell called an oligodendrocyte. That is a big mouthful. Oligo means few. Dendro or dendritic typically refers to branches. Cyte meaning cell. So this is a cell with a few branches to it. And if you can see, these branches extend away from the cell and they contact and form these wrappings around the axon of our central nervous system neurons. And this wrapping around these neurons is essentially phospholipid layers. These wrappings are insulating our axons, much like electrical tape around an electrical wire. In fact, these oligodendrocytes will wrap their phospholipid layer around the axon maybe 50 times. It is a thick wrapping. But as you wrap your phospholipid layers around the axon, all the cytoplasm gets squeezed out into the processes and remains with the cell body. All that's around the axon are just layer after layer after layer after layer of phospholipids and nothing else. But it creates a waterproof, insulated barrier around the axon. And all that phospholipid wrapping is called myelin. And the whole covering along the axon is called the myelin sheath, with little exposed spaces that are called nodes of Ron VA, and we'll talk about those in, probably Wednesday. But you may think, what's so significant about this insulation around the axon? Well, without myelin, an axon transmits a signal at 1 meter per second. With the myelin, the signal is transmitted at over 100 meters per second. So the myelin speeds up the transmission of our action potential down the axon. You know another reason why you can know it's important? What happens if someone loses the myelin around their axons? What disease do you call that? Louder? Multiple sclerosis. Parkinson's is also degenerative. It's neurons in the brain, and we'll talk about that in our final material. But multiple sclerosis is simply losing the myelination. Have you seen people with multiple sclerosis? Their movements are very slow, very discoordinated. The axons are fine. It's they don't have the myelin. So the signals are slowed down. So that gives you a great understanding and appreciation for why the myelin is so important. Certainly in your reflexes. If you didn't have those reflexes moving that fast, you would have serious injuries. So myelin's important. So our 4 cell types, astrocyte, ependymal, microglia, supportive, protective. Our myelinating cell in the central nervous system, the oligodendrocyte, allows the faster transmission of our action potential. Now, peripheral nervous system, we've got 2 that we're going to talk about. The satellite cells, or these pink cells you see in the illustration, that completely surround the cell body of our pseudounipolar sensory neuron. And these satellite cells sort of have an analogous role to our astrocytes, because these satellite cells are going to regulate the nutrients and the waste that are exchanged between the cells and the bloodstream. They're not going to have the little processes that connect with our capillaries, but nothing is going to diffuse in and out of that cell body unless it passes through the satellite cell. Now, the other type of neuroglial cell, and we can see these also myelinate axons, but in the peripheral nervous system, the myelinating cell is a Schwann cell. Now, I saw a new term for Schwann cells in your textbook. Your textbook uses neurolemmacyte. The neurolemma meaning the plasma membrane of your neuron. So I guess these are cells that cover the neurolemma. I like Schwann cell better. I've used it for 30 years. Works for me. But the Schwann cell does the same thing. Wraps phospholipid layer 50 times around the axon, you get a faster signal. So it is the peripheral nervous system myelinator, much like the oligodendrocyte is the central nervous system myelinator. So it's about location. There's that term, neurolemmacyte. That's Schwann cell, means the same thing. The other thing that you notice that's different between these, Schwann cells can only wrap one portion of one axon. Oligodendrocytes, they can myelinate multiple portions of multiple axons. But whether you're doing one or you're doing multiple, the myelin, still phospholipid, still insulating, still increasing the signal. Just use different cells in different locations. So this is just a little bit of how that works. Here in yellow, we can see our axon. And you can see in this case, we've got a Schwann cell. And it's beginning to push its membrane around and around. You see how it's wrapping? That's getting tighter and tighter. And so down at the bottom, we basically have all of the cytoplasmic material and the nucleus in this outer portion. And then there's just ring after ring after ring after ring of phospholipid on the inside. You've seen your parents have an old record of vinyl LP? That's what it looks like, with all the grooves being your phospholipid layers. And in fact, if you look at an electron microscopic picture, that is the myelin sheath wrapped around this particular axon. And I believe this is going to be our Schwann cell. You can see the cytoplasm of the Schwann cell on the outside, and there's the cytoplasm of our axon on the inside. And if you could look at this a little bit closer, you could actually see lines, much like the grooves on your vinyl record. They're called major dense line and interperiod line. Not that you have to know that, but those are the lines that represent either the cytoplasmic or the extracellular face of your plasma membrane, 40 or 50 wrappings as we go around. That's the insulation. That is what's referred to as myelin. All right, so now we've talked about the three structural portions of our neuron. Dendrites bringing in information. You see the arrows. Cell body integrating the information, and axons transmitting or conducting the information away. Now we're going to throw some functional segments into our neuron, and we're going to have 4 of them. The receptive segment, obtaining the information coming in. The initial segment, this is going to be the very beginning of our axon, the axon hillock. The conductive segment, that includes our axon proper. And then when we get to the end, we're going to take those synaptic knobs, and that's going to be our transmissive segment, where we can transduce our electrical action potential signal into a chemical signal, that being neurotransmitters. So now we're going to talk about each of these 4 segments from a functional standpoint and see what they mean. But let's go ahead and set the stage. Let's remember that all cells have a resting membrane potential. And whereas muscle had one that was a little bit lower, minus 70 was the number that I always like to use. And why do we have a negative resting membrane potential? What does that negative mean? That means that there are more negative charges on the inside of the cell. More negative charges on the inside. And there's a couple of reasons why there's more negative on the inside, not the least of which is our sodium-potassium pump. We say more negative on the inside. Certainly we've got all those fixed anions like proteins and nucleic acids. But remember, our sodium-potassium pump's kicking out 3 positives and only keeping 2 in. So we have more positives outside. That's going to make it more negative on the inside, just our pump. But in our neuron, we have these other channels that are called leakage channels. And when we look at these leakage channels, we have some leakage channels for potassium that allows potassium to leak out a lot more than sodium leaks in. So the pump sends out 3 sodium. Our potassium leakage channel lets a lot more potassium, which is positive, leak out. Do you see how that's going to help us stay negative on the inside? All these positives either pumped out or leaking out helps maintain that negative 70 resting membrane potential. Now, once again, when we get to action potential and we would get to sodium voltage channels, what is the threshold? What is that number for our sodium voltage gated channel to open? Negative 55. That's never going to change. Whether we talk about muscle, whether we talk about nerves, that sodium voltage gated channel is always going to open at negative 55 or more positive. So at rest, sodium voltage gated channel is going to be closed. So let's look at our segments. Just again, a little bit of overview to give you an idea where we're going. When we look at our receptive segment, dendrites and cell body, the signals are coming in, stimuli. And typically those are coming from other neurons through a synapse, which means neurotransmitters are going to stimulate the receptors on our neuron. And remember, our neurotransmitter receptors are chemically gated channels. They're either going to open or close, or they're going to signal other ion channels to open or close. And whenever we do this, and we allow those ions to change places, we're going to have what's referred to as a graded potential. You're either going to make the membrane more positive, or you're going to make the membrane more negative, or somewhere in between. And these potentials can add up. So you can either have one big one, or you can have a lot of little ones that add up to be even bigger. And that's why they're referred to as graded. And we'll see what difference that makes when we get into our work probably on Wednesday. Now, the initial segment, which again is equivalent to the axon hillock. This is often referred to as the trigger zone because it's in the axon hillock where the decision will be made whether we send an action potential down the axon or not. Bless you. So all of the stimuli, all of the ions that have changed places in our dendrites, that's added together in the cell body. That's where the integration comes in. Whatever that number is at the axon hillock, that determines whether we have an action potential or not. If we have an action potential, boom. It's transmitted down the axon. It's transmitted down the terminals. And when we get to our synaptic knobs, that's where the neurotransmitters release. We signal, blah, blah, blah. Just like we saw in muscle. So once again, the action potential conduction, the transmissive segment, we've been there, done that, seen that with our muscle. We're going to focus a lot more time up here on these first two. And we'll review these. We'll reinforce and refresh what we got in muscle. Now, how do these different segments do their job? Well, the reason they do their job is because of the different proteins that are present. And these proteins, or the channels that dictate their function, we're going to list them out here. So, dendrites and cell body, the receptive segment. Notice what's here. Chemically gated channels. These are ion channels, potassium channels, chloride channels. There's going to be some sodium channels. But what must you have? What kind of channel must you have if you're going to have an action potential? Say it. Voltage gated. Which ion specifically? Sodium voltage gated channel. Notice what's not there. The dendrites and the axons do not have voltage gated channels. Write that down, circle it, underline it. Because you're changing the membrane potential. And you may change the membrane potential in a dendrite or axon to positive 20. Are you going to get an action potential? No, because you don't have a sodium voltage gated channel. All you're doing is allowing the ions to wash in or out of the cell body. And as that swooshes around inside like a washing machine, all of those ions add up. They collectively change the membrane potential. And whatever that final number of all of that mixture is at the axon hillock, that's going to determine if you get an action potential, because all of a sudden, what do we see at the axon hillock? Sodium voltage gated channel. And so if all of those stimuli coming into the dendrites in the cell body, all those ions changing places, if all of that causes the membrane potential at the axon hillock to be minus 56 millivolts, what's going to happen? What? Y'all all know my tricks, right? Nothing. No action potential. No signal sent. But if all of those stimuli add up and change the membrane potential at the axon hillock to minus 55 or more positive, boom, we get an action potential. That action potential is conducted down the axon because the axon also has voltage-gated sodium and potassium channels, which you have to have for the propagation or the conduction of our action potential. And then when you think to our discussion about synapses in muscle, when we got to our synaptic knobs, we have one additional voltage-gated channel that allowed an ion to come in, and that signaled the release of our neurotransmitter. What was that voltage-gated ion channel? Remember what ion that was? Calcium. Voltage-gated calcium channels. And this is going to be in our transmissive segment in the synaptic knobs. Again, it's going to be the same for all of our neurons. And we already covered that with our skeletal muscle. The only thing that's really going to change for our neurons is what type of neurotransmitter they secrete. So the functions of our different regions are completely tied into the types of channels that are present. Voltage-gated channels throughout, but we only have the chemically gated for our dendrites and our cell bodies. That is important to understanding how we're going to send this information from neuron to neuron to neuron. Any questions before we go on? Okay. So, when we look at the dendrites in the cell body, the receptive segments, chemically gated channels, no voltage gated channels, we're going to see that we have a number of synapses coming in. And you can see some of these synapses are on dendrites, some are on the cell body, but they're all over the place. And so we're going to have a ton, 100 to 1,000 different synapses hitting a particular neuron at any one time. And I want you to notice, some of these they've highlighted in green, some they've highlighted in red. The green ones are trying to illustrate what's called an excitatory change, meaning you make the membrane potential more positive. The red ones are an inhibitory synapse, making the membrane more negative. But all of these reds and all of these greens, the only thing that matters is what adds up right at the axon hillock. So they're going to be graded. They're going to be added all together and integrated in the cell body. Now, since we're talking about neurons and neurons, here's some terms we have to get comfortable with. When we talk about a synapse with muscle, we had the nerve, we had the muscle. We know who we were talking about. But since we have nerve or neuron synapsing with neuron, to distinguish who is who's sending the signal, who's receiving the signal, we name them based on their position to the synaptic cleft, the synapse. So the signaler is going to be the pre-synaptic neuron. The receiver of the signal we call the post-synaptic neuron. Does everybody see how that works? Pre, before, post, after. That's just new terminology we have to put into place. So when we secrete our neurotransmitter, And that binds to the receptor, which is either, again, going to be a chemically gated ion channel or it's a chemically regulated receptor that then signals an ion channel to open or close. We're going to see those ions changing places. And they're going to go down their concentration gradient. If that ion channel is a sodium ion channel, when it opens, where's sodium going to go? In. Sodium's high out, remember? If it's a potassium channel that opens, where's potassium going to go? It's going to go out because potassium's the opposite of sodium. Here's a tricky one. If you're a chloride channel, where are you going to go? You got a 50-50 shot, right? In, I heard in first because sodium and potassium are opposites. Sodium and chloride are always together, right? Salt, sodium chloride. So just remember, sodium and chloride are always together. So chloride would come in. So by changing those positions, opening or closing certain ion channels, that's going to alter the resting membrane potential, either in a more positive direction or a more negative direction. And they're called graded again because they all add together such that whatever the membrane change is at the axon hillock, that will then trigger an action potential. Yes, ma'am. So as you look at, the question is presynaptic, postsynaptic, receiver, and the signaler. If we look at, for instance, in this illustration, do you see the little inset box? It's got the little green sort of excitatory synapse there. Here we have an action potential coming down this axon. When it gets to this terminus, we've highlighted and enlarged the synaptic knob of that neuron. We can see all of our vesicles that are binding to the membrane. This is the signaler. It's secreting the neurotransmitter. That is the presynaptic neuron, the signaler. The postsynaptic neuron has the neurotransmitter receptors, and it's receiving. So the postsynaptic is the receiver, the presynaptic is the sender. Presynaptic is the quarterback, the postsynaptic is the wide receiver. Presynaptic is the pitcher, the postsynaptic is the catcher. I don't know, whatever sport you like. Okay? Got that?